In this video presentation, my group of friends and I will be explaining about the urea cycle. First things first, what is the urea cycle? In amino acid metabolism, free ammonia is produced inside the body. The urea cycle is the process in which these free ammonia will be converted into urea. What is urea? Urea, also known as carbamide, is a colorless crystalline compound which is the main nitrogenous breakdown product of protein metabolism in mammals and is excreted in urine. Why is this urea cycle so important? Free ammonia is toxic to the body and is very hard to be secreted, whereas urea is comparatively not toxic and is easier to be secreted.
There are 5 main steps that takes place in the urea cycle. The first reaction involved is the formation of carbamoyl phosphate that takes place in the mitochondria of liver cell. In this reaction, free ammonia will react with carbon dioxide to form carbamoyl phosphate. This reaction is catalyzed by carbamoyl phosphate synthetase 1 or CPS1, which has been activated by N-acetyl glutamate. 2 molecules of ATPs are required to be dissociated as ADP. The second step is the formation of citrulline. Carbamoyl phosphate from step 1 will react with ornithine in the presence of ornithine transcarbamylase as high-energy phosphate is released as inorganic phosphate. Citrulline then will be transported to cytosol via co-transporter. Just like oxaloacetate being regenerated in Krebs cycle, ornithine is regenerated with each turn of urea cycles. The third step involved in urea cycle is the synthesis of argininosuccinate. Argininosuccinate synthetase will combine citrulline with aspartate to form argininosuccinate. In this ATP-dependent step, carbonyl carbon of citrulline is attacked by the lone pair of amine to produce argininosuccinate. ATP cleaves to AMP, which is adenosine monophosphate and pyrophosphate. Aspartate is important in this step as it acts as amino group donor for the formation of urea.
Fourthly, argininosuccinate layers will catalyze argininosuccinate into arginine and fumarate. The arginine will continue the cycle while fumarate will be hydrated to malate and oxidized to oxaloacetate. Oxaloacetate and glutamate will undergo transamination to form aspartate. This aspartate will be used again in the urea cycle. The oxaloacetate can also be used for gluconeogenesis to produce glucose in the liver. Finally, the last reaction of urea cycle. Arginase cleave arginine to ornithine and urea as the final product of the cycle. Urea will diffuse from liver and transported to the bloodstream of kidney, while ornithine will complete another cycle of urea. Anyways, based on the structure, we can see that the first nitrogen of urea is from free ammonia, carbon is from CO2, oxygen is from H2O, and another nitrogen is given by aspartate that acts as amino group donor. Now, we have a complete urea cycle. Let's see the overall stoichiometry. The equation starts from aspartate and H3, CO2, 3 ATP and H2O, and the products urea, fumarate, 2 ADP, AMP to inorganic phosphate, and finally the pyrophosphate.
Allah mentioned in the Quran, "Eat from the good things which we have provided you, and do not transgress or oppress others therein." Ila al-akhirah ayah, Surah Taha, verse 81. So brothers and sisters, we are all created with such perfection by Allah subhanahu wa ta'ala. He created many things that we can or cannot see, for example, the urea cycle. However, we are the ones who distract ourselves with junk foods, fast foods, cigarettes and such. Because of that, we must put our best of efforts to take care of our body an amana from Allah, and returned it to Allah the same way He gave it to us, with perfection.